So we're back up live with Sarah Chinn in the research lab. Can you describe some of the things here that you work with?
Sure.
While you're...
So this is a setup like for one of the radioimmunoassays that I was using to analyze the sea otter blood samples for things like stress hormones like cortisol or corticosterone. So we've got a setup. I've got my blood samples, and we're using pretty typical laboratory equipment like pipettes, pipetters, pipette tips. We've got our tubes that will our reactions actually happen in, and then different things. So these assays are often, a lot of them that I used are sort of competitive binding assays. So we put in the blood, we put in another antibody that competes with it, or it might be already in the tube if we have special tubes, and then we put in a radio-labeled isotope. And so basically there's a competitive binding process, and essentially it's if there's more of something, it's going to win out with the competition, or with, whatever it's competing with. And so at the end, once the reaction occurs, then we measure what's in the tube. And then from that, we can get a concentration of how much of cortisol or whatever, thyroid hormone, whatever I'm measuring, is actually present in that blood sample. So we're able to get concentrations of all these different markers through doing these assays. And so some of them are also, so we do RIAs, which is what the setup is here. There's ELISAs, a lot of different ways to actually accomplish this with different things like proteins or antibodies or enzymes. And so those are all different assays for different markers that we're looking at.
Okay. Can you explain what an assay is?
Oh, now you're putting me on the spot. It's basically, it's just it's a procedure on how we're going to measure something.
Okay.
And so we call them assays. It's kind of a general term for we have these kits that are already produced by different companies, and they send them to us, and we just put in our blood samples. It's almost like baking a cake. There's a recipe to it,
okay.
And we just add things in the correct order. We give them the right incubation times instead of bake times, and then we can read them later. And we can read them with specialized computer instruments or other readers that can measure whatever sort of enzymes or isotopes or things that we put in here.
Cool. Okay. Then you were just mentioning that sometimes people think about their pets and everything, that, if your pet gets tested for something and it has to actually go out to a lab. But the interesting thing is that you're actually doing that here, correct?
Right. So it's a great skill that I've learned here at Sonoma State, is that I can actually measure these things myself. So I have a dog, but when I take her to the veterinarian, maybe they draw some blood and they want to know, well, how is her thyroid? What's going on in there? Is it too slow? Is it too fast? Or they might send out a blood panel, things like that, to see her health. And I'm actually measuring all these things myself in the lab for sea otters. And so that's a great skill that I've acquired here that actually can be used for any other animal, any other species. Now that I have the knowledge and the technique down, you kind of need the supplies and the assay platforms for that.
Great. And then just one other quick question. It's really easy. Where are the blood samples coming for your study?
Sure.
For sea otters?
Sure. So I was really fortunate. I got a bunch of blood samples from both the southern population and the northern population. So there's a bunch of big minus 80-degree freezers at California Fish and Wildlife, and that's where they're stored. But the people that collected them are from the entire Sea Otter Alliance, Research Alliance. So that includes USGS, UC Santa Cruz, Fish and Wildlife, Fish and Game, UC Davis, Monterey Bay Aquarium in California, among many others, where it's this big effort during captures for all these organizations. They come together. Some people spot otters from the shore to tell the divers and the boat drivers, well, we have groups of otters over here, and then you've got the divers and the boat drivers, and they're actually capturing the otters, and then you've got a whole bunch of people doing other things like working in the vet lab, and so you have veterinarians, you have people supporting the veterinarians, people collecting samples like hair, blood, teeth measurements, body measurements, weight, all these different things. Because once we get a sea otter in hand, we want to get as much data from it as possible, and so from there, I'm using the blood, the serum, and plasma to analyze all these hormones and different markers, and it's really neat. And and I just need a little bit of blood, and I can measure these.
Right, great. I was going to say, and I think it's great that we're using all aspects of, or all populations of sea otters for your study.
And then it's great to collaborate with the folks up north.
Right.
The folks at USGS, they have the freezers up there with the samples, but again, it's the other groups of people, probably Alaska Fish and Game, USGS, Fish and Wildlife, among many other universities as well. And then in Washington, I'm sure other researchers, and then their own state agencies, and everyone working together to get all these samples. And luckily they were in one place, and someone could pick through the freezers, which itself is a very hard job, because I've done it myself. You freeze your little fingers off, but we're able to, from Alaska at least, send me those samples, which is really helps my study. It adds a whole nother level to it, having a comparison between the northern and southern subpopulations.
Definitely. Well, thank you so much for your time, Sarah. We loved having you here today.